We are now on the last sets of PowerPoints. We have Immunology, Proper 1 and Proper 2. In Immunology Proper 1, we will discuss the body defense mechanism. In the body, we have lines of body defense mechanisms, and these are the first line of defense, the second line of defense, and the third line of defense. This is a slide wherein the three lines of body defenses are presented. So we have the first line of defense and the second line of body defense mechanisms considered to be non-specific defenses. In the first line of body defense mechanism, we have the skin, we have the mucous membrane, and we have the chemicals present on the surface of the body. While the second line of body defense mechanism are the processes, phagocytosis, complement fixation, the presence of interferon, inflammation, and fiber formation. While the third line of body defense mechanism, this is now a specific defense mechanism involving the lymphocytes, involving the antibodies, and involving the antigen. So we have here antigen-antibody reaction. So we have here the first line of body defense mechanism and the second line of body defense mechanism considered to be non-specific line of defenses. So we have the first line of defense and second line of defense. And they are non-specific because the immune response of the body could be triggered by any microorganisms or any foreign substances. We all have innate or inborn immunity because in our body we have the innate immune cells that help us fight against any foreign substances or any foreign bodies that will enter into the body. And this innate or inborn immunity is simply referred to as natural immunity. And this immunity is observed in some species of animals and human races. And also, some infections or some diseases are prone to some people while resistant to other people. So just like the white people, because of the thinness of their stratum lucidum in the skin, they are more prone to skin cancer compared to the black people because of the thickness of the melanin pigment in the skin. Some people also are prone to tuberculosis infection while others are not. The mechanical and the physical barriers are part of the first line of body defense mechanisms. We have our tears, which contains lysozyme, and we also have other fluid in the body or body secretions that contains immunoglobulins. Some cells, especially in the respiratory tract, from the nasopharynx down to the lungs, contains surface cells that are ciliated. So it will help the body also to eliminate any foreign substances. The skin also is a physical barrier because of the presence of fatty acids and the presence of normal flora. In the gastrointestinal tract, we have the acidity of the tummy, wherein we have the hydrochloric acid, which also help us fight against any presence of foreign substances. Then we have the urinary system, which flush out toxins in the body through the urine. Okay, we mentioned about the normal flora present in our skin, present in some parts of the body, especially in the gastrointestinal tract. So this normal flora, sometimes referred to as microbiota, are considered to be friendly microorganisms. What are some bacteria that are considered friendly? We have Staphylococcus epidermidis, a small part of the pathogenic staph. We have Staphylococcus aureus. We have the Micrococcus species, the non-pathogenic Neisseria species, and the alpha hemolytic and non-hemolytic streptococci. Okay, what are some of these friendly microorganisms present in the skin? We have the Staphylococcus epidermidis. It has already been mentioned. And we have Propionibacterium acnes. And in the intestine, we have the Enterococcus fecalis and Lactobacillus species. It's found here, Reu10. That is the specific species of the bacteria. So, and there are also other species, Lactobacillus species, that are present. What are the roles of a normal flora? First, the normal flora filled up the space on the skin. So there is bacterial antagonism because they produces bacteriocin, which shut off other microorganisms present, so there will be no more space for a pathogenic bacteria if ever they want to enter into the skin. Another role is nutrient competition. If ever pathogenic bacteria can enter into the skin, there will be nutrient competition because there are already friendly microorganisms present. Aside from the normal flora, we have the cellular components of the body. We have the reticuloendothelial system, or plainly the macrophage system. All of the cells have phagocytic function, so they eliminate any microorganism present. Then we have body reflexes. These body reflexes can eliminate microorganisms presence. We have one, closing of the eyelids. So every time our eyelids closes, it cleanses the surface of our eyes. Then vomiting is also another body reflex that if one will vomit, it will also eliminate presence of microorganisms in the tummy. Then, for the second line of body defense mechanism, we have the body processes involved. What are some of these body processes? We have inflammatory response, we have phagocytosis, opsonization, and fiber formation. There is also an immune process. We have the complement fixation process, which remove or eliminate the microorganisms. The presence of inflammation in the body will indicate other conditions that the body is suffering. What are some of these body processes? We have inflammatory response, we have phagocytosis, opsonization, and fiber formation. There is also an immune process. We have the complement fixation process, which remove or eliminate the microorganisms. The presence of inflammation in the body will indicate other conditions that the body is suffering. There could be diabetes, there could be cancer, there could be cardiovascular condition, Alzheimer's disease, pulmonary diseases, arthritis, autoimmune diseases, and neurological diseases. We have here an illustration of the inflammatory response. So these are numbered into 1, 2, 3, 4, 5, 6, 7 until the elimination of the microorganisms. So here we have the antigens or the foreign substance as the bacteria and other pathogens. Then the platelets are involved if the area is wounded. Then we have the presence of the mast cells that will cause vasodilation of the blood vessels or constriction of the blood vessels. And we have the neutrophils and other macrophages which will destroy the presence of the microorganism until such time that the microorganisms are eliminated. So you just read through the processes involved: 1, 2, 3, 4, 5, 6, and 7. Another illustrations of the steps. Another illustrations of the steps of the inflammatory response. This is also numbered 1, 2, 3, and 4. The same mechanism will apply. The presence of the macrophages and phagocytes in inflammatory process, like for example, the presence of the neutrophils will ingest and kill the bacteria by releasing an enzyme from the lysosome present in the cell to eliminate bacteria or to digest bacteria. The presence of the macrophages and phagocytes in inflammatory process, like for example, the presence of the neutrophils will ingest and kill the bacteria by releasing an enzyme from the lysosome present in the cell to eliminate bacteria or to digest bacteria. There are instances in the body that phagocytosis is not facilitated. So, the target cells or the target foreign substance must contain a marker on the surface of their cell so that they will be recognized by our phagocytes so that the antigens or the microorganisms will be eliminated. This process will release a chemical substance which could be antibodies or complement fragments. And this opsonization usually cannot be facilitated in bacteria that are encapsulated. So they need the process of opsonization. So opsonization is a process by which phagocytosis is facilitated by the deposition of opsonins onto the surface of target cells to be phagocytized. So opsonins are antibodies. So once the opsonins are released, they will be considered as a marker on the bacterial cell, and this bacterial cell can be recognized, so phagocytosis will be facilitated. We, there are instances in the body that phagocytosis is not facilitated. So the target cells or the target foreign substance must contain a marker on the surface of their cell so that they will be recognized by our phagocytes so that the antigens or the microorganisms will be eliminated. This process will release a chemical substance which could be antibodies or complement fragments. And this opsonization usually cannot be facilitated in bacteria that are encapsulated. So they need the process of opsonization. So opsonization is a process by which phagocytosis is facilitated by the deposition of opsonins onto the surface of target cells to be phagocytized. So opsonins are antibodies. So once the opsonins are released, they will be considered as a marker on the bacterial cell, and this bacterial cell can be recognized, so phagocytosis will be facilitated. We have here an illustration of opsonization, also referred to as surface binding phagocytosis. So in the illustration, we have the yellow colored, which is the antigen or the foreign substance. So without the opsonin, the red ones, on the surface of the target cells or of the foreign substance, there will be no phagocytosis. So meaning the cells are not recognized by the phagocytes. So the presence of opsonins on the surface part of the cell or in the surface part of the foreign substance, this will trigger the phagocytes or the macrophages uh, to recognize the target cells. So the antibodies that will be released will react on the target cells and facilitating the process of phagocytosis. So if these are involved, the presence of opsonin are involved, this is opsonization or surface binding phagocytosis. Then we have fever formation, which is the increase of body temperature. So increase in the body temperature These substances are referred to as pyrogenic substances. This could be produced outside the body by bacteria with their endotoxins and inside the body by interleukin-1, an endogenous pyrogen. The presence of fever augments the host body defense mechanism. So we have here an illustration of individuals with a fever. So we have here on the left side a child wherein there is headache. So an ice pack is placed on the head to release the pain or to minimize the pain. And we have here on the right side a person wherein the body temperature is taken. So there is an increase of body temperature indicating the presence of fever. Now, if one has fever, it is an indication that the body is fighting something. We have here a table as a summary of some non-specific components of the first and second lines of body defense mechanism involving innate immunity. So you just read through the columns for the first line and the second line of body defense mechanism. So we have here on the first column barriers and associated chemicals. Then second column, phagocytes. Then we have extracellular killing. We have the complement. We have the interferons. We have the antimicrobial peptides, inflammation, and fever. From the first and second line of body defense mechanism, let us go now to the third line of defense. We have the important process here, the antigen-antibody reaction, and the types of immune response of the body. Immune response is a complex series of cellular interactions activated by the entry into the body of foreign or non-self antigenic substances and biologic agents. There are three characteristics of the immune response: specificity, memory, and amplification. In specificity, it is directed toward and restricted to the inducing agent. So whatever the antigen is, a particular antibody is produced by the body. Then memory, the presence of memory cells in the body will recognize the same antigen on repeated exposure. And we have amplification, the ability to develop and enhance immune response. So if there is antibody formation in the body, we have what we call booster, so that the amount of antibodies will shoot up. We have two types of immune response. We have the innate immune response and we have the adaptive immune response. So these are the normal immune responses of the body. So for the adaptive, we have the humoral immunity and cell-mediated or cellular immunity. Then there is another type of immune response, but this is considered to be an abnormal immune response. We have the hypersensitivity reaction. We have here a table that will differentiate between innate immune response and adaptive immune response. So let us take first the first column, innate immune response. So it is an immediate response initiated within seconds. The target antigens or foreign substances may be groups of pathogens, and there is no memory in innate immune response. In adaptive immune response, the response is gradual, generated over 3 to 4 days, and the target antigens are specific pathogens, and there is memory. Further, this table will differentiate innate and adaptive immunity. So in the first column, we have the innate immunity, general protection, not antigen specific. Early phase of host response to pathogens without requiring prior exposure. The response is immediate maximal response. Does not alter on repeated exposure, or there is no immunological memory. So the innate immunity is rapid, non-specific, and no memory. While the adaptive immunity, highly specific for a particular pathogen, antigen specific, late phase response of antigen specific lymphocytes to antigens, lag time between exposure and maximal response improves with each successive exposure, or there is immunological memory. So in summary, adaptive immunity is slower, specific, diverse, and with immunological memory. In adaptive immune response, the following are involved: antigen, APC, MHC, and specific immune response. What is an antigen? So an antigen is any foreign substance, and it is considered by the body as non-self. The antigens could be present in the environment. So we have the different microorganisms, parasites, bacteria, viruses, fungi, and also in the environment we have pollution and we have toxins. All of these are foreign substances if they will enter into the body, or simply referred to as antigen. Then we have APC. APC stands for Antigen-Presenting cell. It could be an infected cell, and it could be a macrophage. The role of the antigen-presenting cells will involve phagocytosis of the antigen. So we have number 1, number 2, number 3, number 4, 5, and 6 will be the involvement now of the APC. So in number 4, the enemy cell broken into small fragments, or the antigens that are engulfed inside the phagocyte will be broken into small fragments. And the fragments, number 5, the fragments of antigen presented cell on APC surface. So these fragmented pieces will be found on the surface of the cell, and it can be recognized by the body for whatever type of cell or immune response will be involved. So leftover fragments will be released by the cell in the process of exocytosis. So actually, the APC will involve the process of phagocytosis. It will break down the enemy cell or the antigen into pieces, and these fragments will be shown by the cell so that it can be recognized by the body for whatever type of immune response the cell will cause. So we have here in the illustration an antigen which has been phagocytosed, and when the cell are broken into pieces or into fragments, they will be found on the surface of the cell as antigen-presenting cells, and this will be presented to resting T cells, and the presence of the APC will trigger the resting T cells to be activated so that whatever type of antigen present, the activated T cell will form or will produce cytotoxic T cell to destroy the antigen presenting cell. The MHC or major histocompatibility complex or HLA human leukocyte antigen are set of cell surface molecules that code for proteins found on the surface of cells that help the immune system recognize foreign substances, just like the opsonins. In humans, the MHC or HLA are encoded by a large gene family clustered in the same region on chromosome 6. There are two types of MHC or HLA proteins. We have the class 1 and we have the class 2 MHC. The class 1 are found in membranes of almost every cell in an organism or in the body, while class 2 MHC are restricted to some cells of the immune system, to some macrophages, and to some lymphocytes. While class 3 MHC encode for other proteins such as complement proteins, cytokines, and enzymes involved in the immune system. We have here an APC, on the left side an infected cell, and on the right side a macrophage. So the same process involved except for the type of MHC molecule. In an infected cell, we have class 1 MHC molecule, while in a macrophage, class 2 MHC molecule is involved, and the product will be different. For class 1, we have the formation of cytotoxic T cell, and in the other side, we have the production of helper T cell. We have here a helper T cell activation and action wherein APC is involved, MCH2 is involved, CD4 marker on helper T cells is involved, and we have the interleukin secretion and the formation of memory cells and the types of response. So if the neutrophils and macrophages are involved, we have non-specific defense response. And if we have the formation of killer T cell, we have cellular immunity. And if the B cells are involved, they will be transformed to plasma cells forming antibodies. We have the humoral immunity. So we have, for the activation, we have antigen recognition, number one, then formation of helper T cell, then two, clonal selection, formation of several helper T cells, and if the chemical substances are involved, interleukin, it can produce types of responses, non-specific defense responses or immunity. We have cellular immunity and humoral immunity. So in summary, we have two types of immunity. The innate immunity, which will depend upon the genetic factors of the individual, and we have the acquired immunity. The acquired immunity could be subclassified into active and passive. Active if the individual is involved in the production of the antibodies through exposure to the infectious agent, so the individual will get sick, so that is natural active acquired immunity. Or the individual will receive artificial immunization, so after receiving, the individual will produce her or his own antibodies. So this is artificial active acquired immunity. Now for the passive acquired immunity, the individual will be given ready-made antibodies. So we have natural if the individual, example, the baby in a pregnant mother will receive natural antibodies from the mother. So that is natural passive acquired immunity. So it is just readily given to the baby. So the baby is not involved in the production of antibodies. Then we have artificial passive acquired immunity. The individual will receive artificial antibodies taken from other sources. Example, for tetanus, no, the individual will be given artificial antibodies or anti-tetanus so that the individual will be protected. Another illustration of the adaptive immunity, we have the naturally acquired and artificially acquired. So the same, active, passive acquisition of antibodies. So in active, antigens enter the body naturally. The body induces antibodies and specialized lymphocytes. So the body is involved in the production of antibodies. Passive, antibodies pass from mother to fetus via placenta or to infant via the mother's milk. So we have the colostrum in milk, which contains antibodies. Then artificially acquired, active and passive. Active antigens are introduced in vaccines. Body produces antibodies and specialized lymphocytes. For passive, preformed antibodies in immune serum are introduced by injection. So the same as in the other table. So in summary, in this illustration, we have the immune response that can be stimulated by the presence of pathogens or antigens. It could be bacteria, viruses, fungi, and other microorganisms. So the exposure to these pathogens or to these antigens, the body have many lines of defense mechanisms. So we could have the mechanical barriers, we could have the first line of body defense mechanisms, we could have the chemical production in the body or the presence of chemical substances, and we could use body reflexes. Then, if the pathogen is able to enter into the body, the second line of defense will come in, so there will be processes involvement. So we have the chemicals that are produced in the body, we have the cytokines, we have processes as phagocytosis, fever, the production of NK cells, and inflammatory process. If the pathogens still are present, we have the third line of defense mechanism. So this time it is now a specific immunity or a specific immune response. So it could either be cell-mediated immunity or humoral immunity. So if if the individual will survive, of course, life will continue. I would like you to read further on antigen, antibody, types of immunoglobulin, antigen, antibody complex, or reaction, and hypersensitivity reactions, which will be taken up in the last set of PowerPoints. So, from this, I would like to recommend for you to read on these topics. So, the next topic will be immunology proper 2, wherein we have the immune response and the antigen antibody reaction.